All right, well, hello everyone. Thank you so much for tuning into our latest pollution mapping citizen science project findings to date here at the Ocean Research and Conservation Association. My name is Natalie Stevens, and I am a research assistant at Orca and the project coordinator for pollution mapping citizen science. And today I am joined by Dr. Lauren Kleiman, a research associate at Orca who runs our ecotoxicity pollution mapping projects as well. And so today we are going to walk you through all the latest data coming out of this project, including some of the exciting trends and patterns we've been seeing with our annual data. But first, I'd like to start with a brief overview for those of you who are active participants in this project. So the overarching goal of our pollution mapping project here at Orca is to identify pollution sinks and hotspots by assessing the accumulation of nutrients and toxins in both sediment and water samples throughout the Indian River Lagoon over time. And so our sites, which are shown here on these two maps, span a large area of the Indian River Lagoon. And these sites were actually chosen based on a blueprint created by Orca's previous ecotoxicity maps. And some of the sites are actually identified through local concern. And so small teams of citizen scientists go out and collect a variety of both sediment and water samples, which are brought back to our lab for testing. And our citizen scientists who actually participate in the research and gather this data to be analyzed are people of all ages, backgrounds, ethnicities, and scientific levels that come together and help with this science. They go through a formal training with Orca on both the field and lab protocols. Orca staff are out in the field with our volunteers to assist with field work, but our ultimate goal is to have teams be independent in the field, so they'll come to a citizen science center, pick up the gear, and then go out and sample on their own, and then bring the data back so we can do all the lab analyses. And so currently, we have over 100 citizen scientists for Orca's pollution mapping project alone. And these citizen scientists participate in the lab work, in the field work, or in both. And so while I said some teams are independent in the field, Orca staff are always back at the Center for Citizen Science or at some of our sites with our mobile lab to assist with all the lab protocols. And at the very beginning of this project, we only had 3 sites that Orca monitored. But pollution mapping has grown since to 30 sampling sites, and our newest site actually started gathering data in August 2024. And so this project spans well beyond Vero Beach, and we actually have sites across 4 different counties, which includes Brevard, all the way down to Martin County. And all of our sites are located in the Indian River Lagoon and its adjacent waterways. And within our sites, we have 23 small teams that do monitor their site 4 times a year and once during the scheduled month, which is February, May, August, and November, which is actually coming up here soon. And so I just wanted to familiarize you guys with our newest site, that is PM30. It is a spoil island in Vero Beach, actually close to the Round Island boat ramp. And this site is monitored by Vero Beach High School students, and the monitoring funds were actually raised by one of those students, Michaela Bertrand. And this site so far only has 1 data point, and that would be August 2024. And then our second newest site is PM29, and that is again another spoil island, but this time in Fort Pierce. Monitoring for this site began in May 2024, and the monitoring and funding for this site is courtesy of the Fort Pierce Rotary Club. And so as you can see on the screen, there are a lot of different tests that we run once our small teams go out and collect those water and sediment samples. And so this is a comprehensive list of all the tests we are currently running at Orca or going off to our neighboring or partnering institutions. But I'm only going to focus in on a few of them today. And so at the start of each monitoring event, we start with water quality. We measure water quality at each of our sites, which includes temperature, salinity, pH, dissolved oxygen, and hydrogen sulfide. Next up, our teams measure the muck depth and the water depth at their site, which allows us to calculate total depth at all of our sites. Like I said, our volunteers do collect sediment samples, which they'll either bring back to Orca's Citizen Science Lab or to our mobile lab for farther off sites such as in Martin or Brevard County. And there, with the help from Orca staff, we run a variety of analytes. So one of the ones that we go through is sediment particle analysis. And this is where we measure the size of the particles which helps us to note that whether the sites are more silty, which corresponds to smaller and more coarse particles, versus more natural sites, including those that are sandy, rocky, and shelly. In the pore water, we also test for herbicides, including glyphosate. And if you don't know what glyphosate is, it is both a weed and grass killer, and it's actually used by many homeowners. And in this pore water, which is embedded in the sediment, it tends to be higher in nutrients. And this is because the sediment is actually a binding site for nutrient uptake. And we can actually use this pore water as a reliable proxy to what nutrients are actually in the sediment. And in addition, for a subset of our 30 sites, we also collect extra sediment each cycle that is composited and sent off to the University of Florida to test for PFAS, which is otherwise known as the forever chemicals. And so they get this name because once they are in the environment, they are persistent and they do not break down. And there's actually quite a bit of news reports circulating around about PFAS, the negative effects it has both on the humans aspect and then also in our environment. So it's a pretty hot topic at the moment. And in addition, once in November, which we're gearing up for right now, our citizen scientists collect double the amount of sediment, which is sent off to a contract lab in Florida. And there they test for heavy metals in our sediment, including lead, as well as for total organic carbon and total sulfur. And lastly, our teams also collect water column samples, which we take uniformly at elbow deep length. And we're looking for a variety of different analytes, including sucralose, which is an artificial sweetener like Splenda that is resistant to wastewater treatment, and acetaminophen, which is a pharmaceutical drug, which you may know as Tylenol, that is actually removed by wastewater treatments. And we use both of these as our human waste markers as a way to know how much pollution is actually entering the lagoon from either septic systems or wastewater treatment plants. And Dr. Kleiman is actually going to go way more into depth with those two analytes when she is going to be talking in a few minutes. And so our sucralose and acetaminophen data is actually sent to a lab at Clemson University for testing. And we also test for nutrient concentrations in both our pore water and our water column samples. And these nutrients include phosphate, ammonia, and nitrite, which I'm going to talk about in a few minutes. And so obviously this is a lot, and I didn't even scratch all of them, and it results in a lot of data. And like I said, while we do run some of these tests at Orca, we also rely on expertise from other universities and labs, including Clemson and the University of Florida. And I also just wanted to highlight that this list is evolving, and that's based on funding and also what the data is actually telling us. So we've removed parameters, we've added new ones like caffeine in the water column, and we've also are always adjusting it to what is actually relevant for the Indian River Lagoon. But for today, I just want to note that all the analytes that we're going to cover are highlighted in orange, and these are the ones that we're going to walk you through in the next few slides. And so first up, we have our water column nutrient concentrations. And just as a brief overview before I go into this data, when the balance of nutrients in our lagoon is reaching an unhealthy level, this can cause a domino effect of different consequences, including the formation of harmful algal blooms that lead to fish kills. We also see the decline in seagrass populations because there's not enough light actually reaching the seagrasses to grow. And this can also cause consequences across the entire ecosystem. And we can also see this on a human scale, and it can impact our health, which we actually explore through another Orca citizen science project, which is One Health Fish Monitoring. But today, what I want you to focus on this screen is from our latest pollution mapping cycle that occurred in August of 2024. And just as a reminder, this data is only one point in time, so it is just August 2024, and it's just a snapshot in this time. And so if I showed you this figure in February or even May 2024, it would be drastically different. And so to introduce you to this graph, we have the concentration of our water column nutrients for all of our sites, and all of our sites are here on the x-axis. And from left to right, we're also going north to south with our most northern county and sites in Brevard, and then our most southern county and sites in Martin County. And then on our y-axis, we have our nutrient concentrations, which are all represented in milligrams per liter. And then our three different nutrients are also displayed, with phosphate being in the blue bars, nitrate and nitrite being in the orange bars, and ammonia being in the green bars. And here at Orca, we're all about stepping back and looking at the bigger picture or the story that this data is telling us. And so what I really want you to look at is the phosphate. So as you can see, pretty much across all of our sites, phosphate is notably higher than any of the other analytes, so nitrate and nitrite and ammonia. And this is interesting. And especially I want you to see that the phosphate is actually higher in our Martin County sites compared to our Brevard sites. And so again, we wonder what could be causing this? What are the potential sources of these nutrients at these different sites? And that requires us to go back to the basics. And so we explore what major nutrient sources are going into the environment and specifically what this could be for the Indian River Lagoon. And so for nitrate and nitrite, those nutrient sources include stormwater runoff that can carry pet waste and fertilizer. We can see the erosion of natural deposits, since nitrate and nitrite also comes from natural sources. There could be sources such as livestock manure, as well as septic tank waste. For ammonia, we also see some similar sources, but also things like the decomposition of organic matter, like plants. Again, we have that agricultural runoff, animal waste, and that septic seepage, again, tying it to that human waste factor. But if you remember from my previous slide, phosphate was largely the highest of all of our nutrients across all of our sites in August 2024. So I really want to dive into phosphate and what we know about it in relation to our data. And so as you can see here, there are a few major sources of phosphate that I've included and are relevant to the Indian River Lagoon, but this list is not exhaustive. And so phosphate can enter the environment from sources like terrestrial runoff, especially since Florida is actually a phosphate mining state. We again have that stormwater runoff that is carrying that pet waste and even fertilizer when grass clippings are actually getting washed into the lagoon. We have agricultural runoff. We also have wastewater treatment discharge, and again, that leaking septic systems. And this is really important because in excess, too much phosphate can lead to those consequences that I talked about a little bit earlier, including lower dissolved oxygen levels, that blockage of sunlight for seagrass growth, and it can even degrade the habitat for those organisms such as macroinvertebrates. And so with that in mind, I want us to just focus on our Orca phosphate data from 2024 a little bit closer. And so as you can see here, taking just a look at phosphate, we have our values of water column phosphate for all 30 of our pollution mapping sites from the most recent cycle. And again, just a reminder, this data is just one snapshot in time and it's not representative of the annual data, which is coming up next. And so on our x-axis, we have our sites again grouped together by county from north to south. And on the y-axis, we have phosphate concentrations in milligrams per liter. And something new that I'm introducing on this bar graph is these two horizontal lines, and those are denoting the 50th and 90th percentile for phosphate concentrations. And these were determined by the Florida Department of Environmental Protection from a previous study. And so this 50th percentile is 0.1 milligrams per liter of phosphate, while the 90th percentile is 0.33 milligrams per liter of phosphate. And these values and percentiles are actually averages. And what it's denoting is that on the line, it's telling us that 50% of the, that lower line is telling us that 50% of the samples shown here were above this average value from the DEP, while this higher line tells us that our samples are actually higher than 90% of the values that are coming out from that latest Florida DEP study. And so we use these percentiles from the DEP since we do not have any localized percentages for the Indian River Lagoon. But as you can see, our data here suggests that the Indian River Lagoon is much higher. And actually, what I want you to focus in on is that 27 of our 30 sites are actually above this 50th percentile. And you can also see that 17 of our 30 sites are above this DEP 90th percentile. And this has actually grown from our last finding to date, where we only had 11 out of our 30 sites above this 90th percentile. And these sites with elevated phosphate include places like Aqua Arena Marina in Melbourne, as well as Little Hollywood Marina here in Vero. And so again, I'm only talking about August here. And so the next question that we would come up with is, are these phosphate levels this past August similar to the rise we saw in August 2023? And this is where it becomes important to collect our longitudinal or annual data and look at the patterns that we see there. And so here on this graph, I'm showing you a new one. I'm showing you our blocks plot. And this is the trend of phosphate concentrations we are seeing in our water column samples starting in February 2022, all the way to our latest cycle, which is August 2024. And on our y-axis here, we have our phosphate concentrations in milligrams per liter. And on our x-axis here, instead of our sites, we actually have our cycles from, again, our earliest on the left to our latest on the right. And you'll see I also have a color legend here on the right with all cycles February months being in blue, all months in May are in orange, all months in August are in green, and all months in November are in yellow. And so again, I'm using this box plot to represent the annual data, with each box actually representing the distribution of the data from all the sites per cycle. And so this X value is something really important, and that's actually in the middle of the box representing the average concentration of phosphate for that cycle, representing all the sites. And again, this horizontal line is also super important because it's representing the individual box's medians, sorry. But what I really want you to focus on is this pattern, and we've been seeing it across these 3 years of sampling. And so as you can see, we have slightly lower concentrations of phosphate here in February at this mean level, starting to slightly increase in May of 2022, and even more increasing in August of 2022, and then dipping down in November. So if we were to draw a line to represent that, we would see something like this. And what's even more exciting is that we're actually seeing the same pattern in 2023. And then also in 2024. So again, seeing this cyclical trend in phosphate across our sites and across these cycles is really, really exciting, because we're seeing that phosphate is starting to peak in August and then decline in November. And of course, we'll just have to wait for November 2024's data to see if we have that same cyclical pattern. And I also want to show you guys that we did run statistical tests on this annual data to test whether our phosphate water column concentrations were significantly increasing between February and May, especially in 2024. And as shown by these two asterisks, we did get a statistically significant p-value of 0.0015, which does indicate that there is a significant increase in our phosphate from February to May of 2024. And we actually are also seeing this same pattern both in 23 and in 22. And so this just further highlights how important our longitudinal monitoring of our sites is, so we can actually see these patterns and these trends. And while it is difficult to make any assumptions, since the lagoon is such a complex system, we can make inferences on our data based on this, and especially what we know about the surrounding areas where our sites are, such as how close are sites to wastewater treatment plants, sewage systems, even some stormwater runoff areas. And something that we even think about is that potentially that high peak in August could be from the really rainy rainfall season that we have in August here in Florida. And so the main takeaway I do want you to take from this is that, again, it's really exciting that we're seeing this cyclical pattern, and we're continuing to monitor this with our data. And we actually hope in the future to do a comparison of our pollution mapping phosphate annual concentrations to the Kilroy phosphate concentrations. And so now I'm going to switch gears and talk about one of the herbicides that we are testing for in both our water column and our pore water samples. And that herbicide is glyphosate. And so for those of you who don't know, glyphosate is a weed and grass killer used by many homeowners and is actually the active ingredient of Roundup. So it's used in both agricultural and industrial settings, and there are actually over 750 products sold in the US that contain glyphosate. And so there is quite a bit of controversy and debate about the consequences, or maybe lack thereof, of glyphosate on humans and animals. The EPA actually ruled that glyphosate is not toxic to the immune or nervous systems of aquatic biota or mammals. However, the World Health Organization did rule that glyphosate is probably carcinogenic to humans, But again, where that controversy comes in is that the EPA disagrees with that. We also saw that it is not ruled an endocrine disruptor to humans by the EPA. However, we do know that glyphosate has biological effects, and it can be especially harmful for plant growth because it inhibits protein synthesis. And so what is unique about glyphosate is that it does bind to the soil, but again, another point of controversy and conflicting information is the half-life of glyphosate and whether it's persistent for days, weeks, or even months. And so glyphosate concentrations have actually been found by Orca in our water samples, our sediment samples, and even in the fish fillets. And so we are actually one of the few groups that are exploring glyphosate concentrations in all three of these. And as a bit of a fun fact, glyphosate is actually pretty new. It didn't pop up on the monitoring radars for the FDA until 2016. And this just makes our longitudinal data set so much more important to advancing what we know about the concentrations of glyphosate in the Indian River Lagoon. And so what you're seeing here is a graph of the water column glyphosate concentrations of all our sites. And it starts in November of 2022, and then it goes all the way to August of 2024. But right now, we're just going to focus on that year 2022. And so just as a little background of the graph, each site is represented by a line. But what we care about is showing you guys the cycles. So not necessarily the differences between sites, just the differences between cycles. And so there are two y-axes where sites with the solid lines are depicted on the most left y-axis, and then those sites with the dashed line are appearing on the right of this y-axis. And so you might be curious as to why we have two different y-axes. And we do this because of how drastically different the glyphosate concentrations in the water column actually are for our sites. And in order to see all of our sites correctly and to see those patterns and trends, we do need to adjust our units to avoid any of our sites looking flatlined or appearing as zero, even though they are only slightly above zero. And so again, I really just want to focus today on right now on one part of the story, and that is 2022. And you can see this was our first year of monitoring, but we saw a huge spike in glyphosate concentrations. So you can see from February 2022, it rapidly increases, it peaks in about May, and then it rapidly declines up until August. And like I said, we really weren't sure what was happening. The project had just started monitoring for glyphosate. And this really made us curious as to whether the data from year one was an anomaly or are we actually seeing a pattern? And so that made us really curious to see the following year's data. And so I'm going to show you the next year, which is 2023. And what we saw here is really exciting because while we didn't see as huge of a spike, we did see that spike, that increase from February all the way up to May, peaking in May, and then declining into August. And so again, it's nowhere near that high spike that we saw in 2022, but really exciting to kind of see that pattern still going, even if it's at a lower level. And so we again wanted to see what was going to be happening in 2024. And again, we saw a little bit of that same kind of pattern, a little bit more of a delay, it looks like, with not as much starting to increase and peak in February, but starting to increase and peak a little bit more from May to August. And so we're left wondering what actually happened in 2022 to cause that spike. And while we still don't know, it is also reassuring that it isn't happening again, given all the negative consequences we know about glyphosate, and especially that it can have in the environment and to human health. And so our next question would be, okay, so we saw these increases and these peaks across both 3 years, but are those increases significant between all 3 of our years? And so that's where our statistics came in, and we tested for significant differences between February and May for all of our glyphosate data. Again, since if you remember in that 2022, that's when we really started to see that increase in that peak. And so we used p-values and that cutoff again at less than 0.05 to determine if this change and this increase is actually significant or not. And then as you can see, concentrations of glyphosate from February to May did significantly increase with the p-value of 0.002. We did also see a significant increase again from February to May of 2023 with a p-value of 0.005. However, in 2024, from February to May, we did not have a significant increase in glyphosate levels. However, we did see a more, like I said, delayed increase in glyphosate, between May and August of 2024. And again, what caused this later peak is something we're interested in exploring further in the future with our glyphosate data. And it does seem to be that the application of glyphosate was delayed a little bit in 2024. But the main takeaway I want you to take from this data is that glyphosate is present at various concentrations across the Indian River Lagoon, especially in our sites. This is proven by our data. And again, these differences could be from a variety of factors, including rainfall, temperature actually affecting how it's broken down, and then the differences in usage and application. But now I'm going to go ahead and throw it off to Dr. Lauren Kleiman, and she's going to talk about our sucralose and acetaminophen data.
Hi, I'm Dr. Lauren Kleiman, as Natalie mentioned, and I run our ecotoxicity program here at ORCA. I'm going to talk to you a little bit today about different ways that we can identify human influence on the environment. So we've started looking at things called human tracers, and there are a lot of different types of human tracers that we can use, but a lot of them have similar characteristics that you want for them to be good human tracers for environmental samples. So you want them to have prevalent use by humans. You want it to be used a lot by humans so that when we look for them in the environment, you're able to find them. As well as you want them to be able to indicate sewer leakage, overflow, or undertreated sewage, so we can look at some sources of contamination. And if there are studies on the half-life or decay rate of these analytes, it can give us an idea of the time frame of contamination based on how much of it we're finding in a specific area. If we use multiple of these analytes, having the presence of both of those different analytes or looking at the ratio between them can help us to identify different specific sources. And then we want it to be specifically linked to humans. So we don't want to look at a medication that's maybe being applied to pets and could be detected in pet waste at the dog park or agricultural animals. And we don't want it to be something that's going to be fully metabolized by humans. We want it to only be partially metabolized so that it is passed on into those wastewaters that will then be passed on into the environment eventually. So the human tracers that ORCA is currently looking at, although we're starting to incorporate caffeine as well, are sucralose, which is, as Natalie mentioned, an artificial sweetener. This is resistant to wastewater treatment. So the influent going into the wastewater treatment plant or the raw sewage coming in can have very high concentrations of sucralose, and some of it might be broken down. You'll probably have reduced concentration in the effluent from the wastewater treatment plants, but it will still be found in those environmental samples in that effluent and in the water coming out of the water treatment plant because it's not fully broken down by that treatment. And there have been some identified negative effects on some fish species and different microbial communities from sucralose, but those are generally at much higher concentrations than what people are detecting in the environment. For acetaminophen, This is a pharmaceutical commonly found in Tylenol, but also present in over 600 other drugs, so very highly used by humans. And this is useful to us because acetaminophen is removed by wastewater treatment. So while you'll see very high concentrations in the wastewater influent into the treatment plants, it should be mostly or all the way removed by that full wastewater treatment, so that when we're finding high levels of acetaminophen in the environment, it can make us think about faulty septic systems, sewage overflow, sewer breakdowns or leaks, and that can help us indicate some of these sources of contamination. Acetaminophen also can impact fish development and reproduction, but again, this is at pretty high concentrations, which aren't what we're finding currently in the environment. So we wanted to look at some previous studies and see what's been found about some of these tracers in order to compare it to ORCA's data on these human tracers. So for sucralose, in these previous studies, they measured the concentration of sucralose in the influent going into our wastewater treatment plant. And those studies had mean concentrations of about 40,000 parts per billion, 48,000 parts per billion. And then in the environment, remember, this is not being removed by those wastewater treatment plants, although it may be reduced. In these previous studies, the sucralose is being detected at a majority of the sites that they're testing, and the maximum concentrations are still pretty high. So one saw about 37,000 parts per billion, 27,000 parts per billion, 12,000 parts per billion. So still pretty high concentrations, and those tend to be higher nearer the effluent from these wastewater treatment plants. Comparing that to ORCA's data, so our data, we have a partner at Clemson University who analyzes this data for us, and we were able to send them samples just right now from Indian River County due to our funding sources. So this was funded by Indian River Community Foundation and Schmidt Family Foundation to be able to look at some of these samples. So right now, our data that I'm going to be showing is just Indian River County, and we were able to send samples from our August 2022 cycle up through about half of our February 2024 sampling cycle for a total of 96 samples. And we detected sucralose in 88 of those 96 samples. So similar to those previous studies, a majority of our sites were showing this sucralose. Now, our maximum concentration, about 1,700 parts per billion, is much smaller than the maximums in some of these other studies, and our mean, similarly, is fairly low, about 334 parts per billion. If we look at acetaminophen, in previous studies, in the influent or that raw sewage, raw water that's coming in for treatment, acetaminophen was consistently detected in that influent, and the mean concentrations were in different studies were about 78,000 parts per billion, about 122,000 parts per billion, so very high concentrations coming in. And then a lot of that wastewater treatment is removing most or all of that acetaminophen so that in the environment, it's being detected at a relatively small number of sites that are being tested, and the maximum concentrations are much, much lower. It was only 11 parts per billion in one of the studies, and then around 380 parts per billion in some other studies. When we compare this with our ORCA data, again, those same 96 samples from Indian River County, our maximum concentration of acetaminophen is only around 55 parts per billion, with a mean of around 2 parts per billion, because we had so many samples where we weren't detecting them. And our maximum is right in the range of or below the maximum in these other studies, which were conducted in, I think, mostly New York. So different system, but we're seeing much lower concentration, at least in our samples. Again, these are just snapshots, which is why we're trying to look over time. So we're able to use a ratio of these two human tracers of the amount of acetaminophen to the amount of sucralose in a sample. And that ratio can tell us something about the source. So in a previous study, the ratio of the influent for the wastewater treatment plant is greater than or equal to 2.5. And coming out of the wastewater treatment plant, this is only calculable when you have a detectable amount of acetaminophen. The average ratio was very small, 0.009. And so we're able to look at these ratios in the environment and try and tell at what point our contamination may have occurred. And this can be a little bit complicated because this ratio does decrease over time as the acetaminophen can degrade in the environment, especially exacerbated by higher temperatures and microbial activity. So we have our 2.5 going into the wastewater treatment plant, 0.009 coming out of the wastewater treatment plant. In our ORCA data, we only had 15 samples. We detected both sucralose and acetaminophen. So when we calculate those ratios, our max is 0.19 and our mean is 0.05. So both of those are falling between that full treatment of the wastewater treatment plant. So what this could indicate is that there was some contamination by acetaminophen at some point, but it's had enough spatial distance or enough time where it's degraded enough and we see this decrease in that ratio. A lot of our sites had a lot of variation, either between sites or even within sites, where you could see acetaminophen detected at one cycle, but then most of the other cycles it wasn't detected at that same site. And that can also tell us some things about the contamination, as if there was something like a faulty septic system, you would expect to see more consistently high concentrations of both of these analytes. So we're really excited moving forward with this data about what it's going to be able to tell us as we're able to gain funding to hopefully look at sucralose and acetaminophen in our other counties, as well as moving forward in time and being able to dig more into specific sites that maybe had some of those higher concentrations and look at why that might be. For our next steps, we're expecting to get back some more of that PFAS data from our water and sediment samples for the past year, back from our collaborator at University of Florida. So we're really excited to see what that's going to show, especially since this is such an important topic right now, as we're seeing a lot of health issues from PFAS. We're also starting to test for microcystin in pore water. So that is the biotoxin produced by the blue-green bacteria harmful algal blooms. So we'll be able to look at that for pore water samples. As I said, continuing our acetaminophen and sucralose and continuing adding in caffeine. And then moving forward in our November sampling, where we do those additional annual analyses, we're going to add in iron. So we're able to start looking at some things that might be correlating with iron and affecting some of the pollutant cycling in our sediments. With that, we want to give a huge thank you to the many people who helped fund this project. A lot of them help volunteer with us as well, but we are a nonprofit. We can't do any of this without you, and we really appreciate all of the many people and community organizations who believe in this project. We're starting to have enough data over time and spatial distance where we're really starting to find some really interesting trends. So it's really exciting for us. We hope it's exciting for you. And a huge thank you also to our citizen scientists. Without them, we couldn't do nearly as much as we do. As Natalie said, we're going from Titusville down to Martin County. We have a bunch of teams that are currently independent, so Orca has a pretty small staff. We couldn't do all of this field sampling, lab analysis, and be able to do as much solid, reliable science as we are currently able to, thanks to our many dedicated volunteers. And we just have a few references for some of our numbers and statistics. And you can feel free to reach out to Natalie or myself with any questions about this presentation or the project or how to get involved. So thank you very much for coming to our presentation.
Thank you, guys.